Okay, hello everyone. Thanks again for being here today at my workshop about the analysis of spatial transcriptomics. So what are we going to do today? So think about that. Someone in your lab or whoever, a friend, made an experiment using some sort of spatial transcriptomics platform, and what was generated as output was this a table like this, a table with literally millions and millions of rows filled with some numbers. And now the question is, okay, what do we do with that, right? How can we make sense out of these spots, out of that table, and how can we get sort of an idea of what are we actually looking at? So the goal of this workshop is to turn something like this, a table like this, into something more visual, and clustering and gene enrichment analysis and things like that. So it's going to be a hands-on workshop. I will guide you through a very basic Python pipeline on how to do these kind of analysis steps. But before we start with that, I just want to give sort of 15 minutes of a presentation about spatial transcriptomics and Merfish in general, specifically Merfish, because that's what we're going to use today. By the way, if you're not using Merfish, but something more like 10x Visium, it's not that different from an analysis point of view. And I'm just going to give you sort of a brief overview on that part to make sure that we all are sort of on the same page on that. I'm going to talk a little bit about data processing, and then we will start with some programming or some data analysis. So what is spatial transcriptomics? I'm sure most of you know or have at least a pretty good idea. It was actually named Method of the Year 3 years ago, which seems like recently, but also seems like a long time. There's been a lot of things going on in that field. And it's interesting for me because at least here at Broad, whenever I'm talking to other researchers, independently sort of what they're researching on, very often the discussion goes into that direction that they also want to try some sort of spatial transcriptomics to get more insight in their data. So this is really a big topic in the last few years, and it continues to grow. So basically, spatial transcriptomics, it's spatially resolved transcriptomics, right? We want to add spatial information to the RNA sequencing. So instead of having these classic single cell experiments where we generate these gene expressions, we now additionally to that also want to have sort of a spatial understanding where these cells are. And I found this nice analogy in the internet, sort of the evolution of single cell spatial analysis. And it essentially started with bulk RNA-seq, right? So imagine we have this population of cells, a tissue, and we are interested in getting insights about the sequencing information in that tissue. And the first thing that was done, that was like broadly used, was this bulk RNA sequencing. And in bulk RNA sequencing, you look at sort of groups of cells, right? So you group your tissue together, and then you look at these individual groups. And this works great, and you can read a lot of information out of that. But you end up averaging over multiple cells, right, over all these groups. So the information that you get is not super it's averaged, right? It's not very specific. It's not single cells. And so obviously, the bigger these groups are, the sort of more averaged out or like more general the information is. And then if you decrease and decrease and these groups and you get smaller and smaller groups, you have more precise information about your tissue. And if you go on doing that, essentially you will arrive at a group of with only one sample, and that's essentially what single cell RNA sequencing does, right? So we here are not only looking at we're not anymore looking at groups of cells, we're not averaging anymore, but we are looking at specific cells individually. However, to do that, similarly to bulk RNA-seq, you essentially destroy the tissue, right? The tissue is dissolved, and you then have information about all these individual cells, but that's about it, right? You don't know anything else. You don't know where in the tissue these cells were. You don't know the neighbors of these cells, etc. And this is where spatial transcriptomics comes in. Spatial transcriptomics aims to add spatial information to it. So instead of like looking at these individual cells sort of in a row, in a list of cells, we want to look at them as a graph, right? We want to understand, if you look at this one specific cell, what are its neighbors, what is the relationship to these neighbors, and that really just adds a whole other dimension to this to RNA sequencing, and this is essentially what makes spatial transcriptomics so powerful. And so spatial transcriptomics, essentially it has sort of two schools of thought, right? So there's sequencing-based spatial transcriptomics, where, as the name suggests, you essentially do single cell sequencing of your tissue, but before you do the sequencing, you add a spatially resolved barcode to it, such that once you're done with your sequencing, you can then map that single cell back to an XY coordinate in your tissue. And the sort of opposite approach on that is what's called imaging-based spatial transcriptomics. And this is where you're not using this classical sequencing approach, but instead you're using different fluorescent microscopy technologies and protocols to visualize the transcriptome within the tissue. And most notably, the platforms, especially I think everyone knows 10x Visium is the most well-known sequencing-based spatial transcriptomics, and I think still to this day also overall the most used platform in spatial transcriptomics. And then in imaging-based, you have, for example, Merfish, which is commercialized by VisGen, and we are using Merfish data today for our analysis. And then these days, 10x actually also has an image-based platform. But of course, for both of these, there are many competitors. There are different platforms out there. So very briefly, what are sort of the pros and cons of these different ways of doing spatial transcriptomics? So sequencing-based, because it's sequencing-based, it gives you the full genome resolution. So it gives you the whole, you do sequencing. So you have all the information that you can get with single cell sequencing. And the drawback of that is that you have limited resolution. And that is, I'm not going to go into the very specifics of that, but you have sort of a capture area, which is essentially an array where you keep these barcodes that you use to map your single cell sequencing back to a coordinate. And this resolution of barcodes is essentially limited. And what that means is that if you look at these dots, these represent the spatially resolved barcodes. These are actually not single cell resolution. So it's usually multiple cells aggregated together. And imaging-based methods, on the other hand, they have usually they have a limited amount of genes. Very often you cannot image the whole genome, although with Merfish you can record tens of thousands of transcripts of genes. That kind of depends on the specific platform that you're using. But the interesting aspect of image-based protocols and technologies is that you usually have subcellular resolution, right? So not only have you the information about every single cell, but you even have a higher resolution than that. So you see where inside the cell, where are these individual transcripts that were detected. What is Merfish? So Merfish stands for multiplex error robust single molecule fish. It was developed or published in the year 2015 by a group around Harvard researchers, I think Harvard Medical School, and then Jeffrey Moffett from Boston Children's. And so their idea, or the main idea of that platform, is that you have multiple rounds of imaging, and the genes are assigned to a binary code. And this is sort of the multiplex approach. And then what's really fascinating about that platform, I think, is the error robust aspect of it. And that comes from the fact that these binary codes that you assign to a gene, they're error robust. So if you switch, so these are like a 16-bit vector, so 0, 1s, length with 16 bits, and then if you flip a single bit, it will still give you the correct genome. So I want to give you a little bit more of an insight or a hint how this is working. So here you see a bunch of sort of abstract RNA transcripts, A to G, and they're assigned a binary barcode. And then in each imaging round, typically, or the protocol is used with up to 16 imaging rounds, but you can, of course, use fewer imaging rounds if you want to. In each round, you decode a single digit, a single binary digit of this barcode. So in the first imaging round, you decode the first entry of the vector, and then you do this single molecule fish, SM fish, which allows you to probe the transcripts for a specific molecule, and you use fluorescent imaging to see whether the probe is there or is not there. And so you see this 0, 1s, and that corresponds to what you will read out. And as you can see here, once you go on, right, you add these other bits to it, and at the end here with three rounds, you have a binary code for each transcript. And again, what is meant by this error robustness and by the error robust code is if you would switch now one of these bits here, this transcript might not be detected as an E anymore, but you can be sure that it will not be wrongly classified as a gene A. That's the error robust nature of it. So how are we doing that, right? Essentially, we have these imaging rounds, usually 16. So really, what you get, the raw data that you get of your experiments is a sort of gigantic stack of images, right? Grayscale images, fluorescent images. Sometimes you even have a Z-axis, so many images and lots of data. And this is not super interesting to us because what we actually want is sort of these gene expressions, right? These gene expression matrix. So how are we doing that? And this is not really going to be the topic of today's talk, but I just want to give you a sort of a hint. So there's data pipeline doing that, which involves multiple steps. So it starts with sort of bringing these individual field of views that you have, bringing them together, combining them to sort of a mosaic image of the whole tissue. Then you need to do the spot calling. So you look at these individual spots, right? In each round, you analyze the brightness of these spots and you say, okay, this is a 1 now. And then you combine them at the end, and at the end you assign a spot to a gene, or you say, this is a mistake or whatever. And then typically that these pipelines also involve segmentation. And this is actually something I think more complex than what we are going to do today. And there are some open source tools, namely there is Merlin, which can only be used for Merfish. Merlin was developed originally by the same lab who also developed Merfish, but it's not maintained anymore, and it's not really functional. I mean, you can make it work if you tinker around a little bit. It's not terribly broken. There are some versioning issues with packages. And then there is Starfish, which was sort of a bigger approach funded by CGI to build an open source software that supports various different platforms. But that unfortunately didn't really got too much traction, and it's also not maintained anymore. But as far as I think, as far as I know, it's still functional, but it's not maintained anymore. And this is, I think, sort of a big challenge in this space of spatial transcriptomics, that there are not enough and not like not good enough open source tools out there to do this data processing. And so, again, what we are doing today, going back to my first slide, is to turn this table into something more useful. And we are going to use something that is called SC-Verse. I'm sure many of you already heard about packages like ANData, ScanPy, or SquidPy. These have all been developed from the same community called SC-Verse. And they really built over years the fundamental tools for omics data. And you will see that in a minute, that these are really great tools. They're very well documented. They come with a large number of functionality. So clustering and plotting is already implemented, and you will see how we're going to use them. But this is really going to make our lives much easier when doing data analysis. And this is kind of in contrast to what I said before. So when we are talking about data analysis, there are actually a lot of really good tools out there, namely these. But when it comes to data processing, not so much anymore. And not 100% sure why that is, but I think that a lot of researchers also then they purchase the platforms from a vendor, 10x or VisGen or whatever, and then that usually comes with analysis software. So these steps that I briefly mentioned here, in my opinion, probably most researchers don't use these open source tools, but go straight for the proprietary software that is provided by the vendors. Nonetheless, of course, I think it would be very good if I had a better collection of open source tools to support that. So the basic building block of this data analysis is ANData. And ANData, you can sort of see an abstraction here, is a data object specifically built to represent omics data. It works with single cell sequencing, but also as we are going to use it with spatial transcriptomics data. And in the center, sort of, you have this, what is represented by an X. These are the spatial coordinates of your transcripts or of your cells in our case. and then you add variables and observations and things to these individuals spots. And we're gonna see in a minute a little bit. I think that will be a little bit more clear. And just here another sort of illustration how that is built up. So really we take as input whatever platform you're using, and we construct these and data objects. And we are then using these other tools from the SCVerse, ScanPy and SquidPy, to build spatial graphs, to do clustering. And then on top of that, if you would want to, you can interface that with other tools, with machine learning, etc. So the data that we are going to work on today is an open source data set that was published along this paper here. Most notably, it's also Jeffrey Moffett is one of the main authors, and he was also on the original Merfish paper. So he's one of the sort of inventors of Merfish. And you can find all of the data here if you want to have a look. What is actually what I want to mention here is that I mentioned previously that in theory with Merfish we have subcellular resolution, right? So we can see the individual RNA transcripts within the cell. However, for most these analysis steps, we then aggregate these to a cellular level, and that's what we're going to do here. So the building block that we're going to use for data analysis are the cells that have been segmented. In that case, they have been segmented by this Bayesian segmentation algorithm, which you can find it's described in this paper. And then we calculate the gene count matrix for each of these cells, and that's what we use as the basics for our analysis. So if you are interested to learn more about that, this is a great review article on spatial transcriptomics. It's already 2 or 3 years old, but it's very comprehensive. They have a supplement material, which is literally the size of a book. It's like 600 pages. It feels like it lists every single package you might want to use or might consider using. So it's very comprehensive. They also have a good, I think, Jupyter book that you can go through. There is this single cell best practice. This is also a Jupyter book, which gives you a great overview on the SCVerse and what you can do with it. And it provides a lot of basic tutorials for you to get started. And if you want to know more about spatial transcriptomics in general, I can really recommend this YouTube presentation. It's from a group in Sweden, and they go over I think like the 5 most well-known spatial transcriptomics platforms, so including Merfish, single molecule fish, etc., in a little bit more detail. So if you're interested in understanding how that actually works on a biological and chemical level, then I can recommend watching that talk. And so now let's get started. So for today, I'm providing either a GitHub repository or a Colab notebook. I'm gonna share the link in a Zoom chat. Oops. Did you receive the link? So this is a Google Colab notebook. I prepared everything such that in theory you should be able to run it step by step. But of course, I'm going to give you instructions on how to do that. Is that link working for you?
Yes.
Okay. And alternatively, if you don't like Colab or you prefer to do it yourself, I also have created a small repository which contains an environment file, so you can go here. I'm also going to share this link, and you can create your own conda environment and then follow the steps on your local computer. Okay. I think it's probably the easiest if you use the Colab notebook. You can always go later on and clone this notebook. Okay, so yeah, let's get started with sort of the more hands-on part of this workshop. I think it's best if everyone, so I mean, if you don't want to follow step by step on your own, feel free to just watch and let me explain to you what I'm doing and what's going on. But if you want, it's great if you also follow on your own in the Colab. So before we do that, does anyone have any questions so far? Okay, so for the hands-on part, feel free to interrupt me anytime. It's hard, I think, doing a workshop. It's hard to estimate sort of what's the knowledge of people. So if I'm going too fast, let me know. Also, of course, if I'm going too slow and you're bored, let me know. Yeah. So first step for those of you who want to go through it on your own, just try to run the first few cells of the Colab. The very first cell is simply importing and installing a bunch of packages. However, that sometimes fails. Yeah, exactly. If it fails, you can try just to rerun it. It often works the second time for whatever reason. Yeah. This step is sometimes a little bit slow. So are people following along or are more interested in just me going through it?
Trying to follow along.
Okay, great. Thank you. Yeah, so if you're here at this step, so again, if this the first cell breaks, simply rerun it. I don't fully understand why that is, but if you run it the second time, it usually works. And then here at the mounting step, it will ask you to allow access to your Google account, Google Drive, but that usually should be fine. Otherwise, if you're not okay with that, feel free to check out the repository. And then this is just downloading the data. I prepared that as a zip file. We download it and then it's ready to use for the rest of the notebook.
So can we only use this notebook for Merfish data, or can we also use it for other kinds of spatial transcriptomic data?
So the analysis part, once you create a AND data object, can be used for other platforms as well. However, the import, and I'm going to briefly mention that the import is specific to the platform that you're using. So if you're using Merfish, you need to import the data in a certain way. If you use 10x, you need to import the data in another way to end data.
Thank you.
Okay, let's get started. This is exactly, thanks for that question. What you mentioned. So as I said, we're gonna mainly work with these SC-verse tools, ScanPy and SquidPy. ScanPy is, so both of them, they work interchangeably because they are built on top of AND data objects. And then ScanPy provides functionality for single cell sequencing, and SquidPy for spatial transcriptomics data. And we start by importing the data, and SquidPy provides data importers for the most common platforms. So they have, for example, an import for VisGen, they have an import functionality for 10x, and I think for many more. And it makes it very easy because you just do sort of ScanPy.read.visgen, and then you provide the paths to where your data is. However, for what we are doing today, this was not generated by a VisGen device. So we are going to sort of import it manually. So the first thing that we're going to do is importing the data frames that have been generated by the segmentation. And what we need in almost any sort of spatial transcriptomics analysis is two things. We need the gene count matrix. So for every segmented cell, we want to have this vector which counts how many entries of gene A and how many transcripts of gene B were found inside that cell. And that's what's generally referred to as the gene count matrix. So this is the first thing we import here. You can see that on these are the individual cells here. They're numbered from 0 to I don't recall how many cells it is. And then these are the genes here, alphabetically ordered, and these are the counts, right? So cell 1 doesn't. Cell 0 doesn't really seem to have a lot of transcripts, but then when we look at these other cells, they really have a lot of transcripts of this specific gene. And that's the first thing that we need. Now the next thing is that we now need to have the spatial information, or the information about where the cells are. This is what's usually referred as the cell metadata, and we load this into this metadata data frame. This contains is, it contains again the sort of the index of the cell, and it then contains XY coordinates of these cells, and a bunch more things. Here, these are how many transcripts were found in general, and then you have how large the cell is, etc., etc. And the tricky part to some extent is that these data frames are different depending on how are you segmenting your tissue and how are you calling your spots. So if you're using VisGen, so if you're using the software provided by VisGen or the software provided by 10x, you don't really need to worry about that because they will make sure it's consistent, and you can import it very easily with these existing importers. But for data that is generated by sort of custom made or non-proprietary algorithms, you then sort of need to stack that a little bit more together, and you need to be a little bit more careful about how the things are named, right? So here it says this is the cell. Sometimes it's called a cell index. This is sometimes called slightly different. This could be called X global or whatever. Here you have something like average confidence. I'm going to talk about that briefly in a minute. That could be present or it could not be present depending on what decoding and segmentation algorithms you're using. Okay, let's get started by something that is very often done. So we are very much interested in clustering, but then also sort of in looking at where are specific genes enriched and how much are they expressed in specific regions of the tissue. And sometimes, depending on your tissue, you have genes that are present or that are expressed similarly across the tissue. So you could have a gene that is very highly expressed, but it's very highly expressed in every single cell found in this tissue. And then that's not very much ideal for what we are trying to do, because it's in a way, it's sort of like adding noise to our analysis pipeline. It seems to be really, really important because it's very highly expressed everywhere, but at the same time, because it's highly expressed everywhere, this makes it not very relevant for our analysis. And so one thing that is very commonly done is that you define a list of genes that you want to drop, that you want to filter out, because you assume that these genes are not relevant for your analysis. And in our case, we actually don't have any genes that we want to filter out for that tissue. And this is also something, right? I mean, this is a decision made by the data analysis pipeline, right? This is not, obviously cannot be defined globally, right? It depends on your tissue and on your experiment, and maybe you don't want to do that, but it's something that is very often done and helps to improve the results of your downstream analysis. So once we done that, or in our case, we're not really doing that because this gene is actually not present in our tissue. We can then go and build our data object. And this is this single line of code here. What we're doing here is we assign the gene counts, the gene count matrix. This is going to be the centerpiece of our analysis. So there was a question. So do we do this list manually depending on the tissue organism? Yes. This is depending on your experiment, on your tissue organism, whatever exactly. Yeah. And yeah, as I said, you can also leave it empty if you want to. Yeah. Yeah. So going back here, if I quickly go back to sort of the overview on N data, you have this X object sort of in the middle. This is the gene count matrix. And then you add these observations and variables corresponding to these entries in that matrix. And we can see here that we are defining the gene counts as the X, and then the cell metadata is becoming the observation, right? So this part here. So what is very sort of almost like the coolest thing about N data in general, and we're going to see that, is that this is now the object where all our data lives. And in contrast to many other analysis pipelines for different packages, like different things like imaging or whatever, we are actually never gonna extract information out of that. We're only gonna add information to it, and you will see that in a second. But this is in my mind the most interesting part of that this package. You can see here the observations that we have added. We have a cell index, we have the XY coordinates of the cell with the number of transcripts, density. So these observations are coming from this data frame that we imported up here. And I'm going to talk about this a little bit more. I think it will get clearer in a minute about what I try to explain here. Yes, so we also going to add, we already added the coordinates here. So the first thing that we're going to do is to do some basic pre-processing steps. So now we have successfully imported our data. Just to make sure, is there anything unclear? Any question so far? Okay, we'll go on then. So, okay, the data is imported. The data is living in our AND data object. And so the next two things are two very basic pre-processing steps. And the number one is filtering. So I mentioned previously that, you know, we are building up on top of a segmentation pipeline, of a segmentation algorithm. And segmentation obviously isn't always perfect. And if you have a large tissue, you will end up in a situation that some cells have been incorrectly segmented. And that means that, for example, what's happening in spatial transcriptomics very often is that you actually don't have a stain for the cell membrane. So you always have a stain, you always typically have a DAPI stain for the nuclei, but very often you don't have a stain for the cell membrane, which means that that makes segmentation difficult, and it can happen that essentially you end up segmenting only the nuclei, missing out the large portion of the cell, and therefore sort of discarding a lot of transcripts that are actually part of that cell. And for example, one thing that can happen is that you end up having a cell that has no transcripts assigned to it. And this is what we try to sort of resolve with our first pre-processing step. So we're going to define two important variables. First thing is the min counts per cell. This is the minimum number of counts required for a gene to pass filtering. So what that means, if we have a gene that has not been so I think that was you said that incorrectly. 1 second. Oh yeah, sorry, I said that incorrectly. So okay, the mean counts per cell, what that is it's the minimum number of transcripts you want to have in a cell. And we are setting it here to 1, and what that means is that we are filtering out every cell, where every empty cell, right? If a cell has no transcripts assigned to it, we're not going to continue to use that cell, we will discard it. And I think that most of you would agree that's a sensible thing to do, right? We want to do single cell analysis, and if a cell doesn't have any genes in it, you know, there's nothing to be analyzed with it, so we can just throw it away. In a very similar way, we also going to define minimum cells per gene. And what that is, it might be that you have a gene that is only present in like 2 cells. And then you can ask yourself, okay, maybe there's something wrong with that gene. Or it could also be that in your decoding pipeline, you know, it was more difficult to sort of decode that specific genes. It could have multiple reasons, but you also very likely want to discard that gene. And so in this specific example, we're setting the minimum cells per gene to 3, which means that if a gene has less than 3 cells that it was detected in, we'll throw it away, right? So we are only going to carry on with genes that have been detected in at least 3 cells. And this number is actually still pretty low, and you know, both these numbers are of course parameters that need to be set by whoever does the analysis. And again, I think it depends very much on the experiment. So next thing, sort of first real analysis part is quality control. And again, this is one of the reasons why these SC-Wars is so great, because they already provide functionality to do quality control, and all we need to do is to run the functions and specify some parameters. And specifically, we are interested in 2 metrics that correspond to quality control. One is called total counts, and the other is called n genes by counts. So both these metrics are going to be calculated for each cell. And the total counts is means the total count of decoded transcripts or spots for a specific cell. And the n genes by count is the number of genes with at least 1 decoded transcript inside that cell. And so that might not sound super intuitive. So I came up with this beautiful abstraction of a cell. So in this specific cell, we have decoded 3 different genes. We have this purple gene where we have found 4 transcripts. We have the orange gene with 2 transcripts, and then we have the green gene where we found 1 transcript of it. And so the total number of transcripts decoded in that cell is 7, right? This is just summed up all the transcripts. So the total counts is equal to 7 for that specific cell. And the n genes by count is 3, right? We have 3 different genes with at least 1 decoded transcript. So it might be that actually in our experiment there's also a red gene or a blue gene, but these genes have not been found inside that cell. And so I want to just show you quickly how the data, this N data looks right now. This is what we started, right? I sort of talked about that. And now we're gonna run our quality control. So let's run it. And now what's sort of what I tried to explain earlier, what makes this N data extremely nice is that if you look at the difference up here and down, is that after we ran quality control, you now see new variables in our data object. And this is really the key idea of N data, and this is what makes it so nice. We are not sort of taking this object and then running through an algorithm, and then that algorithm returns to us the number of these total counts per cell or the N genes by counts, but the functionality calculates these values and then adds it to the object as additional variables or observations. So with each of these steps of this pipeline, we continue to add more information to our end data object. And whatever is present here, we can later on use for plotting. And so this is a really nice aspect of this end data objects. This makes analysis very convenient, right? You don't have now a lot of variables floating around in your notebook. It's all present within a single object, within a single data object. And we can always go and like reference now these n cells by counts, etc. So this is nice, but we calculated these values. I said we want to do quality control, but that in itself doesn't really tell us anything. So the next step is that we want to plot that. We want to sort of obviously have an idea how good the data is. So we want to visualize the quality control. And so I prepared these basic histogram plots here. And just to, again, to follow up on what I just said, right? You have now in this end data object, you have all these variables, right? You have the mean counts, you have the n counts, you have average confidence area, etc., etc. And you can now use these variables inside all sort of plots. And here we create a plot with 4 different histograms. And for example, let's have a quick look at the first histogram, right? We simply use Seaborn here to plot a histogram. And then the beauty is that now we just say, oh, this is our end data object. We want to take one of the observations. Specifically, we want to take this total counts, which was calculated by the quality control step and then was added to the end data object. And in a similar fashion, here at the very end, at the very beginning, when we imported our data, I quickly mentioned that the segmentation algorithm often provides the area of the cell as well. So we can do the same thing, right? We can go, oh, please also plot a histogram of the areas of the cells, and then a histogram of the average confidence of the cells. And so that then results in these plots that give us a rough idea of what is going on in our data. So these are sort of the most, I would say, useful metrics for doing quality control. So the average confidence, that's not outputted by every single algorithm. Essentially what it is when you do spot calling, you get an idea of like how confident are you that this specific spot is belonging to a certain gene, right? And so usually almost all these spot callings algorithm provide then a confidence value. And very often you would also filter your data for these. So you would say, oh, if the confidence is like 60% that this is it's a transcript of gene A, you will probably throw it away and only keep the spots with very, very high confidence. And what you can see in this histogram plot is that the vast majority of data actually has a confidence of more than 80%, which is good. So that already gives you sort of quality assurance about the data, right? If that were like skewed here and you would see sort of a Gaussian like that, you might want to reconsider what you're doing here on if the data was processed correctly. Let's have a quick look at the total counts. As I said, this is the total count of decoded transcripts per cell. And it's quite common that this is like sort of skewed to the left with a lot of values near 0. So there are a lot of cells where you only have a small number of transcripts. And so this, I think, looks pretty good. I think from what I know, it always kind of looks like this. And then this is what we mentioned up here. So how many genes were found in a specific cell. And what you can see here is that this is sort of almost nicely Gaussian distribution, right? Which is something you would expect, that there's sort of a mean how many genes you have per cell, and it's sort of distributed around there. And again, here, right, if you would have high values in the first interval, that might be something to worry about, right? But looking at this, seeing this nice distribution, I think can give you some confidence that this data is good to go. And again, you can try to plot pretty much whatever. So whatever you can find in here, whatever is present in the end data object, you can plot in a histogram. Do we have any specific questions to that part of the analysis? No. Okay, let's go on. Yeah, similarly to so here up here we have created these histogram plots. We can also go and plot this data in various different ways. We can make a violent plot, whatever. So for some values, it might not be enough to look at this histogram and then be like, oh yeah, that looks great. But you might want to have a better understanding of the distribution, and so you might want to plot something more like a bit more sophisticated visualization of your data, for example, a violent plot. And again, here, ScanPy provides this functionality already, right? Which is just super convenient. You do scanpy.pl stands for plot, .violent plot, and then you tell it whatever you want to plot. So this is the end data object. You specify the variables that you want to plot. So for example, if we remove this one, it's only gonna plot the first value. And then here you have a bunch of parameters that you can set, and you can look at them yourself and kind of play around with it if you want. So next step after quality control usually is data normalization. There's not really much to talk about it. I think it's pretty much straightforward. You normalize some of the values. Might have not really sure if I understand your question correctly. Haven't you lost your raw counts if you don't put them into Merfish raw?
Yeah, so the versions I've seen online, you have to do assign .raw to your original X table, because when you do the normalized, the X value or the X table is going to get replaced by the normalized and log value. So you've now lost your raw counts. I didn't know if there was something I didn't know about where it was keeping a shadow copy of that somewhere.
Yeah, I don't know. That's a good point. Yeah, that might be a good point. Yeah, I'll have to look into that. But thanks for that. Yeah, maybe getting that to full step. Okay, So moving on to something a little bit more exciting, I would say clustering. Again, I want to give you so I think to answer that question, maybe better, at least one of the normalization was added as a separate variable. So maybe we should put in place to false, and then it's going to add another variable to it. But you're probably right that if you put in place to true, that it's going to replace the values that you had. Yeah. So one of the nicer thing about these packages is that they also provide a vast collection of clustering and sort of clustering algorithms, PCA, UMAP, Leiden clustering, etc. There are many more. So if we run that might actually sometimes takes a few seconds. And again, here it's pretty much up to you to whatever clustering you want to use, right? They have UMAP, PCA, they have TSNE, and they also have various sort of similar hierarchical clustering algorithms. Yes, it's finished. And then again, I just want to emphasize what I mentioned already a couple of times. If we now look at the data object, what we can see is that once again, all of what we calculated here was added as another variable to our object, right? We have now here the XPCA, we have XUMAP, and so we have the UMAP clusters and the Leiden clusters, etc. And this is really sort of the key in my mind of this package, right? This just makes it very, very easy to use that because traditionally, and if you would do something with images, it would look something like this, right? You calculate your clusters and then it returns you something, and then that would be of whatever data structure size. And whenever you want to do that, when you want to plot, you need to then look at how that was returned, and you need to plot it, etc., etc., etc. And the idea of this whole SCVers is that they really provide you all of that functionality in sort of one set of tools. And this is exactly what we're going to see here. We added this UMAP here, right? So what we can do is, again, scanpy.plot.umap will now plot the UMAPs. And exactly. So we now not only, so you can actually specify what sort of what observations and what variables you want to calculate the UMAP dimensions on. Maybe I think for that, it's most interesting to just look at the clusters, and what you can see is that UMAP now came up with 16 different clusters. It's been visualized here. And again, I think this is just like, let's take a moment to sort of appreciate how easy it is to do clustering and then plotting of such data. It's literally like 2 lines of code, right? So that makes our life much easier. No, I don't really have any tips or suggestions to choose the parameters in Leiden, unfortunately. Yeah, that again kind of depends on the data. And I mean, these clusterings, to some extent, this is what's nice, like PCA, principal component analysis. It's not used that much anymore. It was sort of replaced by UMAP and TSNE. However, when you do PCA, first of all, I think it doesn't have parameters, right? Yeah, but also you know what it's doing. And all these other clusterings, I'm actually not sure about Leiden, but for sure with TSNE, right? There's a lot of randomness in it, and you need to be very careful with these clustering algorithms. So for example, with TSNE, you actually can take random noise, so dots that were generated randomly, you can throw it in a TSNE algorithm and it will give you perfectly looking clusters. So it will attempt to make sense out of nonsense. So yeah, these clusterings, you know, it's always something to be a little bit careful or skeptical with. But no, unfortunately, I can't really give you a hint on how to choose that. Yeah, so yes, and then so what we did here is we now plotted the clusters, right? Visualized in the UMAP dimensions here, right, in 2D. But of course, it's much interesting to actually see how these clusters are looking in the spatial dimensions. And so we can define to plot these clusters not on the UMAP dimensions, but on the spatial dimensions that we added. And that's how this then looks like. And, you know, this is where sort of it goes into a little bit more the biology. So full disclosure, I'm actually a computer scientist. So, you know, whenever you sort of move from doing statistical things or sort of analysis steps, and then you want to answer the question, okay, does that actually make sense? That then becomes a little bit more difficult and really depends on your experiment and whatever you're actually doing. So, but if you look at that, at least you can see that, for example, the screen cluster, the second cluster number 2, is distinctly, it seems like it's distinctly present in here. And then, so you can see that the clusters are sort of spatially separated by plotting it there. But, you know, to answer that question, or the question, okay, do this cluster now make sense? That's not a trivial question to answer, and we will see a little bit more going into that. So again, just very easy to plot these things, right? Very convenient. However, the only thing that is less convenient is that this gives you a static plot, right? This is amazing to just give you a plot of that in a single line of code. But if you want to do a publication of your data, or you want to change certain things about these plots, or you even want to have sort of an interactive plot, you need to do a little bit more work, because then you know these static plots aren't you getting anywhere, and there's only so much you can do to change how these actually look like. And so here I'm presenting you sort of an example on how you can turn that into an interactive plot using Plotly. I think this is not really worth going into in much details. If this is something you're interested in, feel free to later go and have a closer look at it. Essentially, what we're doing is we are extracting the information out of the and data objects. So we are getting the clusters and we're getting the spatial coordinates. And then we are just building a plotly graph that is interactive, right? And we are setting the spatial coordinates as, and we add a bunch of configurations and settings to it. And you end up with an interactive plot, which also means you can sort of zoom in, right, and hover over the things. And so this helps you to get a better understanding of the data and how it's helps you to inspect the data, basically. Yeah. So, any questions so far to clustering or plotting? Nope. Okay. So yeah. What did we do so far? So we started with importing our data frames. We did some basic filtering, removing some cells that are useless, some genes that are useless. We did some quality control. We now have confidence, sort of, that what we are using makes sense. It's not complete garbage, right? Have high confidence values. We have nice distributions of our data. And we also did some basic clustering already. And so now let's go a little bit further, and let's now try to understand a little bit more about these clusters, right? And so these clusters, or the way, or sort of this image, is essentially also the reason why you want to do spatial transcriptomics, right? This is the difference to single cell sequencing. So these individual dots are the same, sort of the same output that you get from single cell sequencing. And now the difference and the amazing thing about spatial transcriptomics is that you now actually can map them to an XY coordinate. And so apart from like plotting a pretty image of it and make it look nice, what makes that interesting is that we can go a step further, and we can now ask the question, okay, we have these clusters here. We have a cluster here, we have this cluster, etc. They're sort of spatially resolved. They spatially probably represent some sort of cell types. Now, can we map that to, can we map a cluster to a cell type that is actually known in biology, right? And can we, or let's ask the question, how is a certain gene expressed in a certain cluster? And the first thing that we can do here is to plot a gene ranking. So for each cluster, we can plot like what are the most important genes within that cluster versus all the other clusters. So we now take, I think this is a nice example here. So we take cluster 1. We took all the cells that are assigned to cluster 1, and then all other cells who are not assigned to cluster 1. And we do a correlation analysis, and you can specify the method that you want to use. So you can, for example, so in this example, I'm using a Wilcoxon test. You can use a t-test. You can use linear regression. Again, ScanPy doing a great job, basically implementing all sorts of statistical correlation tests already for you. So you can just specify the method. And what's happening now is that for each gene, it will calculate the sort of the correlation between the gene and a specific cluster, and then it will rank them. And this graph, just to make it not too busy, I'm plotting the top 10 genes. So the 10 genes with the highest correlation to a specific cluster are shown in here. And what you, for example, can see here is that in cluster 1, we can really see that there's one specific gene, this Vim gene here, that is expressed very heavily in that cluster, but not so much in the other clusters. And we can see similar things in cluster 2. But then if you scroll down, you know, we have clusters where we don't really see some genes really sticking out and then like giving a hint, sort of like, so all of them are relatively flat. So then that kind of raises the question, okay, if you know, all genes are sort of equally important, how did you cluster it, right? And probably, so it could be two things. It could mean that we have too many clusters and it's sort of just at some point split a cluster into two that actually should belong to the same cluster. But it could also mean that for cluster 10, there was actually not a cluster that was generated for a specific gene expression, but maybe for a specific region. So that here, for example, for 10, it might be that this spatial information was more important to cluster that. So that in itself is not, it's not super useful. It just gives us an idea. I would argue if all the clusters look like this, you know, you should probably reconsider your clustering algorithm and try to change the parameters. So I personally think about that. I think about this plot as, you know, some sort of another step of quality control, but this time a quality control of your clusters. So what we can do next is we can actually now plot the correlation of the clustering with specific genes. So what we're doing here is we are plotting the principal components, the two principal components, and then we are visualizing two genes. And I actually selected them not randomly, but if you see here, this is this Vim gene, which you can see here is very much expressed in a specific cluster and not so much in some of the others. And we have yeah, why do some have slight lower scores while having higher ranking? Okay. That's a good question. I'm not sure if I can give you a correct answer. I think it depends sort of how, like what method that you're using, because the clusters are not built exclusively with these gene expressions. So there you have these gene expression matrix, and then you have the spatial information. So you have some dots in your a dot with an XY coordinate, and then that dot is essentially characterized by the gene expression vector. So it's not necessarily that two clusters are different because they have different gene expressions, but they can be different because they have different spatial locations. And I think that's why you sort of end up in a situation where what you described, where you have where you can have like higher rankings but like lower scores. And these these rankings are also sort of, this is always like one cluster versus the rest of all clusters. Yeah, I hope that sort of, did that clarify it somehow, or?
Yes, it did. Thanks. Like you can see from the 5 versus the rest. So hi from Germany. So I was just asking about if you see the NRP6, the genes, and the besides it, because you have this kind of clusterings. So maybe it's the method that involve only a certain criteria and not the spatial, like you were saying.
Mm-hmm.
Okay, that's
Yes.
all right. Okay. All right. Thank you.
Yeah, thanks for that question. I think this is really a great question because it sort of gives you a hint, right? Like this notebook is nice and it's kind of like Python code, and you can visualize stuff. You can plot like correlations and expressions, but at the end of the day, it's very important to what you just said, right? To try to make sort of biological sense of these things, and it's not trivial to do that. So it always depends on your experiment or on your tissue, and then depending on that, you might want to choose different clustering algorithms, different parameters, and for example, different methods on like how to rank these genes. And what makes these analysis steps hard, or like I guess in some sense what makes biology or bioinformatics hard in general, is that there's no definitive answer, like what cluster to use and what parameter to use. Yeah.
Is there
Thanks for
any
that
rules or the thumb rules or something that, because we do have a lot of genetics, genomics data and proteomics, is there any kind of rules that we can use, for example, when we're using Merfish analysis, or is just I can try anything?
Yeah, you can try anything. No, there are
okay.
not really any
Specific
rules.
rules. Okay.
No,
All right. Thank you.
Thank you. Yes. Yeah. So yeah. Okay. So here, just very, very simple, sort of looking at these plots, we can see in the one versus the rest, there's one gene here sticking out. We can see, where was the other? Yeah, we can see in the 0 versus the rest, sort of kind of sticking out. But what's interesting is if we now plot the principal components of all our cells, but then we visualize the expression of specific genes. And that's what we did here. And what you can see is that we do see very clear differences between these two genes, depending on the principal components of the cells. So this again, sort of in itself is not very useful, but again can give you somewhat a hint about how well your clustering worked, right? And again, I would probably argue if these would look the same, right? You might reconsider your clustering. And to see a difference in the principal components when you mark two genes that you know should be expressed very differently in different areas, then you also should assume that you should see a difference in the PCAs. So that's a good thing. So I want to move on to a little bit more interesting plots. So here, similarly, we plot again our cells on the spatial coordinates. And like here we plotted them on the PCA components. Here we go back to our XY coordinates. And we now do something very similar than to here. We again plot the expression of some of the genes. And this is actually, again, sort of emphasizing the need or the purpose of spatial transcriptomics. Because what you can see is that for this this gene, right, that is very much exclusively highly expressed in a specific region of the tissue. And that probably makes some sense, and in itself might not be surprising or exciting. But this is something that you cannot do with just single cell sequencing, right? And this is sort of, again, this is kind of the purpose of spatial transcriptomics is exactly that, right? That not only do you know the gene expressions, but you know where they correspond to in the tissue, and then you can actually do these kind of visualizations, right? You can visualize the gene expression within the tissue, and you can try to see differences. Where is the gene expressed differently within the tissue? So yeah, this is sort of a very basic example on doing that, but again, sort of emphasizing spatial transcriptomics. These are, I don't really want to go into it. Maybe very quickly, you can create what's called a spatial neighborhood graph. So you take your tissue, and now you create a graph out of that. So you link the cells with its neighbors. That allows you to, again, for very nice downstream analysis later on. So now the spatial neighborhood graph in itself doesn't really do anything. But what we can do, what you can do now is we can sort of plot a heat map of this spatial neighborhood, which is called neighborhood enrichment. And so again, we created a spatial graph, which is essentially just taking this spatial image, this XY coordinates of our cell, and then specify, okay, for one cell, what are the neighbors of that cell? And that's how you build a graph over all of our tissue, by connecting these cells with each other. Now, an interesting question is actually, how are these clusters spatially, how are they spatially enriched? And ideally, you would say, okay, if a cluster should, if two cells with the same clusters are close together, right, they should be enriched. Or if two, or the other way around, actually, if two cells are close to each other, they should actually be most likely of the same cluster. And we can give an idea sort of like plotting a heat map like that, where you expect the diagonal to have a very strong signal, giving you the idea that cells that are close to each other in this graph, in this neighborhood graph, are also likely to be assigned to the same cluster. So I want to move on to the sort of last part of the analysis, which I find is also the most exciting part. And it's essentially we can define marker genes. So I already said I'm not a biologist here. So for the biologists under you, this might be super trivial and might be not as exciting as it is actually for me. What we can do now is we always know sort of what kind of tissue we have, right? We know what tissue we have, and then as a biologist, we know sort of what cells should be present in that tissue. For example, in our tissue, we have muscle cells, we have T cells, we have stem cells, we have TA cells. And as biologists, you also know better than me that very often such cells can be characterized by a small number of genes that are very highly expressed specifically in that cell. Doesn't mean that they're not present in other cells, but they're significantly lower expressed in other cells. And what we can do now, and this again, sort of to follow up on an earlier question, this again is completely depending on the experiment and on the tissue that you're doing. And this is sort of where biology then meets bioinformatics, right? This needs to be sort of, this is a biological input sort of into our data analysis platform. So what we are doing here now is that we define a number of cells that we know from a biology point of view that they should be present in our tissue. And then we define, for each of them, we define a list of genes, a list of marker genes. Doesn't need to be a list, right? You can only have a single gene, you can have 10 genes, whatever you want. And we can use this now to see if there are, or we can use this to try to identify a cluster as a specific cell type, right? So we used sort of unsupervised clustering for our data, and now we actually want to make sort of biological sense out of that. So we ask the question, okay, we have 16 clusters. What do this cluster actually mean? What we have done before is sort of looking at, okay, how much sense do this cluster make, right? Here we looked at, okay, if cells are close to each other in space, are they close to each other in terms of how they're clustered, which is true very often. We also have looked at the sort of gene rankings, which gave us a very rough idea maybe about what these clusters actually mean. And now we're going to go a step forward, and we want to be able to say, okay, cluster X corresponds to stem cells, and cluster Y corresponds to T cells or whatever, right? And what you can do is, once we have defined our sort of marker genes that correspond to specific cells, we can plot a histogram like this. So, and what you can see here is that we defined this gene here as the marker gene for smooth muscle cells. And what you can see is that this gene is very strongly expressed in cluster 2, but very weakly expressed in all of the other clusters. And that's essentially giving us a hint and saying like, hey, okay, there is a high chance that actually cluster 2 corresponds to these muscle cells. And a similar thing actually holds for the T cells, right? We have defined 2 marker genes, and we see very, very strong expression of these marker genes in a specific cluster. And the same for the stem cells, right? And you see for the stem cells here that they're very strongly expressed in cluster 0, but then they're also sort of medium expressed in other clusters. So, you know, that can change, like, depending on like how many genes you actually use as marker genes, what genes are you using, etc. And so this is nice. So we can now maybe take the assumption, okay, cluster 2 is muscle cells, cluster 6 corresponds to T cells, and we can plot that. Let me see. So here again, just once more emphasizing how easy it is to plot things, right? We are plotting and embedding on the spatial coordinates, we want to visualize the clusters and we want to group them. So these are the groups. These are the cluster names, which in our case are numbers. And now we want to visualize two specific clusters, namely that is the cluster 2 that we think corresponds to muscle cells. And then it's the cluster 6, which we think might correspond to T cells. And let's see what's happening. Yeah. So that then allows you to sort of backtrace our assumptions that we can take from these marker genes and from these heat map to try to have a look at that tissue and maybe give us an idea of how much sense that actually makes. There is a question. Okay, so the question is, I'm not actually sure if people can see the chat if I open it. So the question is, in your experience, the definition of these marker genes, do they go in line with those used for single cell sequencing analysis? Or do they need to be redefined for spatial analysis? So no, they don't need to be redefined. So the marker genes are really a biological thing, right? That they don't really have anything to do with whether it's single cell or bulk RNA or spatial sequencing. These marker genes, I would say, are usually coming out of like what is known about biology, right? So if you have a specific cell type like T cells, they have been sequenced previously in single cell sequencing, and so you sequence T cell and then you sequence other cells, and you see this big difference in gene expressions, and you say, oh, okay, this gene XY can be considered a marker gene for this specific cell type. So no, it doesn't actually matter. It's a biological sort of question. What do you take as a marker gene for a specific cell? Yeah, so I'm not sure why it's not. No, okay. Yeah. Let's see here. What else do we want to plot? Here we plotted 6. Let's fix 0. Oops. Yes. So here, for example, I think this plot is kind of nicely looking. We plotted cluster 2. So looking at this heat map about the marker genes, cluster 2 corresponds to the muscle cells. And then we plotted that versus cluster 6, which corresponds to the T cells. And then so, you know, again, these plots in itself are not necessarily telling you like, oh, like this is not like ground truth, right? These plots are there to either help you better understand the data, help you to sense check the data, or sort of give you more insights in the data. Another question. Is there a way to analyze microenvironments? Could you maybe explain what you mean by microenvironments? So maybe I'm not really sure what you mean by microenvironments. If you maybe refer to like sort of a small subset of the tissue, Yeah, in that case, yeah, sure, you could just like crop that out and do your all these analysis steps specifically for a certain smaller region, or if you're using interactive plots, you can just zoom in. But I'm not very sure if that's what you meant. Yeah. Okay. Yeah. So these plots or these steps again. Okay. Maybe another question: is there a way to import a list of cell IDs to subset the regions? There a way import the list of cell IDs to subset the regions and compare subregions using the same or overall clusters from the initial analysis? Okay. So is there a way to identify specific neighborhood with different cell types compositions? I'm sure there is a way. I don't think that there's like a built-in function that you can easily just plot that. So you will probably need to go and write some custom Python code to do that. I would say, yeah. So no, I don't know a way to do that, at least not with built-in SEOs ScanPy functions. But I'm sure you could somehow do that. But yeah, again. Okay. Yeah, again, this sort of allows us to attempt to plot different cell types within the tissue. And then just like you can also always vary sort of the plots that you want to do. So here we started with a very basic heat map, but you can use different plots. So once again, ScanPy really provides various different plots. Instead of doing this simple heat map like above, we can do this dot plot that gives you a little bit more information. Or we can also do this sort of classic heat map. So whatever you prefer. So these are both just other example of plotting literally the same thing than above, just to give you an idea about the diversity of sort of plotting functionalities that we have. Yeah, so with this, I'm actually at the end of what I have prepared so far. I'm happy to take any question. Feel free to clone the repository. You can find the notebook that we just looked at in here. Let's go. You can find that here. I'm providing a conda environment file, so you could just conda install that. That actually, I think, contains most packages you would need for analysis. I also provide additionally a notebook. Oops, okay, that's too big. That's unfortunate. That notebook provides some code how you can actually read read and import the raw data. So the DAPI stains or the cell membrane stains, and how you can visualize that data, if that's something you're interested in. Okay, the micro environment analysis, I mean something like k-means neighborhood. Yeah, I think, let me see what you mean. I think, yeah, I think you could do k-means, but I haven't tried, so I'm not really sure if that's something you can do, but I think that they have it. And then, yes, another question, how do you know of any spot calling algorithms that can decode dual fluorescent label objects as honestly, no, I don't. Merlin, not, I would say. Maybe have a look at, what was it called again? I forgot the name. Maybe have a look at Starfish. I think Starfish used to be the most sophisticated one that supported a bunch of different protocols. However, it's not maintained anymore since like maybe like 2 years. So if it's a newer protocol, I doubt that that will be supported. Yes, so in general, yeah, that's a very good question. And I think, once again, I want to emphasize that when we are talking about the analysis of spatial transcriptomics, we really have an amazing collection of tools, especially with the ScanPy and SquidPy things. There are also others. So there's a lot out there that help you to do sort of this kind of data analysis. When it comes to spot calling and segmentation, it's just a different story. There is no sort of like standard on how to do that in terms of open source. And again, I think it's partially because most researchers are probably using the vendor software that comes with the device. Yeah, but yeah, unfortunately, no, there's no software that I'm aware of that helps with that. Follow-up question. Instead of spots that you're plotting, can you plot it as segment whole cell? Yes. So actually what we are doing here is the whole cell. We reduce that. So that's done very often in the analysis step, is that you look at it in the cell level. So whatever you see here as a spot is not a transcript spot, but it's a cell. And if that were to be 10x data, the individual spots here would be groups of cells, right? You have this in 10x, you have this microarray, so you don't have single cell resolution. So that would be sort of aggregated cells together. Yes, but these are cells. Yeah. Yeah, thank you. Yeah. Okay. Any other question, remarks? Okay. Okay, there's one more. Yeah. Okay, then I think I'm 10 minutes early, so that's good. At least I didn't run out of time. I want to thank you all for coming and for attending this workshop. I really hope that it helped you somehow. Yeah, and yeah, thanks very much again for attending, and thanks for your attention.
This was really great. Thank you so much.
Thank you. Yeah, thank you very much. Okay, goodbye, everyone. Thanks again to all of you.